Think is pretty general across a lot of different clinics. Um, and what we see here is that this is the trajectory of speech recognition development in postlingually deafened individuals after they get their cochlear implant activated. And what you can see here is that the trajectories in this figure are really variable, both in the rate at which word recognition accuracy grows and the ultimate performance that they asymptote at. So some people do great. So you got people up at the end of like 90% open set word recognition accuracy. We consider that a really successful outcome when you're listening with a cochlear implant. But you've also got patients at the bottom end of the scale who start with nothing and after 2 years of using the device still have almost nothing in terms of speech recognition ability. And this is really unfortunate because from a clinical perspective, if what you're trying to accomplish is preoperatively counsel your patients as to what's going to happen after they get this major surgery, um, they don't care about what the average outcome is for their patients. They care about what their outcome is going to be. And without a good ability to either make sure after the fact that we can ensure that good outcomes occur or preoperatively make judgments as to who is likely to benefit and how large that benefit is going to be, it leads to a situation where you're going to have a lot of patients who are falling in the bottom of this end of this range and end up really struggling to make the most of what their device is theoretically capable of giving them, and can lead to a lot of disappointment and regret around this. So what I'd like to do is to try and figure out what the mechanisms underlying this variability are, and for the things that we think we might be able to fix in the clinic, figure out what are the pathways to be able to make modifications to their devices to improve those outcomes. So my lab's approach to this problem takes two angles, and I'm going to break this into two halves of the talk that I'm giving. The first half is going to focus on auditory access to speech cues. And I've been developing methodology specifically to look at how much speech information people can extract from different frequencies across the speech spectrum, and also look at in listeners of cochlear implants, the response of the auditory nerve to electrical stimulation and how variable that is across patients. The second half of the talk that I'm going to give is going to focus on cognitive factors that we think facilitate speech recognition. And the two factors that I'm going to focus on that get a lot of attention in the literature are attentional control and what I'm calling verbal memory. So the first thing I want to start with is anchoring our talk of access to auditory speech cues in the current clinical gold standard. So the Speech Intelligibility Index is a measure that we use to try and predict how good somebody is going to be at speech recognition, given either an adverse listening condition that they're in or whatever their configuration of hearing loss is. And it's also used to try and design assistive devices like hearing aids to try and improve access to that speech cue and maximize a person's ability to extract speech cues from what it is they're hearing. So you'll have to forgive me. I am an engineer by trade, and so I'm going to have a few equations throughout my talk, but I'm going to try and talk through the plain English of what these equations mean, more so than focusing on the technical details themselves. But the way the Speech Intelligibility Index is defined is that you're looking at, for every frequency band in a signal, you're calculating how audible the signal is in that band, and then you're multiplying that by how important that information is. So best case scenario, if a frequency band is highly audible and it's important for speech recognition, that'll contribute substantially to your ability to understand what you're hearing. Conversely, if you can hear a signal in a specific frequency band well, but that's not important information, it's not going to help much. Or if it is important information, but it's not audible to you, you're also not going to get much benefit to your ability to understand speech from that part of the signal. And so we use this equation to predict at sort of a population average how well somebody's going to do at speech recognition. And the work that I'm going to show next is trying to extend this to look at individual differences in particularly the weighting function that we see here. So the current standard for how this is calculated usually produces results that look something like this. The way we do these measurements of the importance of these different frequencies for speech recognition traditionally is you will do some filtering to the signal. Here I've just drawn a mock low pass filter where we're removing all the information above about 3 kilohertz from the signal. And we assess speech recognition accuracy. So usually we present words or sentences through this filter and ask people to just repeat back whatever it is they thought they heard. We look at how accurate they are at understanding that speech. And then we move the filter. Here I'm moving it down a bit, and we're then asked them to do the task again. And we compare the difference in performance across these two conditions. The idea being, if we just filtered out information that was really important for speech recognition, your ability to understand what you're hearing should go down. Conversely, if we remove some information from the signal and your performance is unchanged, that means that that was probably not something that was important that we removed from the signal. And when we do this across a bunch of different frequencies, we get a trend that's fairly general across stimuli, although there's some idiosyncratic properties of specific speech stimuli. But on average, what we get is that this 1 to 2 kilohertz frequency region tends to be the most important. So we often see a peak there. And then at the edges of the spectrum, the lowest and highest frequencies tend to be less important. That's not always true, but listening in quiet, this is often the pattern that you'll observe. Now, this is really useful clinically because this gives us targets for how we program, especially hearing aids. Like we know if we can amplify the signal around 1 to 2 kilohertz to make it audible to a patient with hearing loss, they're probably going to do better with that hearing aid than if we have a hard time amplifying the signal to make that audible. But the problem with this is we can't really do it on an individual basis. So the traditional way of making these measurements is really, really time intensive and tends to be done at a population average. So you'll get, 100 people that all have similar hearing profiles, have them do the task, and then look at that group average. But I think that's not necessarily good enough, especially if we're looking at patients with variable configurations of hearing loss, because what we really want is to be able to make these measurements on an individual basis to say what it is that any given patient is using to understand speech. So to address this problem, I and a few other people in the field have been working on revising the methodology that we use to make these measurements of importance. So here, instead of doing this progressive high pass and low pass filtering, we design stimuli. Here I have 4 example words with their spectrums, where we're sort of randomly cutting out which frequencies you can hear or not hear in any given trial. And what we do then is we have more modern statistical techniques where we can look at what's the effect of having every band present or absent in the signal. And we can do this much more efficiently by making these sort of random combinations of frequency bands that people hear in the task. So at the smallest case, we can do these measurements in 5 to 10 minutes if you want a really coarse function, like 6 spectral bands. And if you want more bands, this jumps up to maybe hour and a half of data collection. So it's probably something that's still not clinically feasible, but for experimental purposes, we can start to look at individual differences in performance and importance for speech recognition using these sort of revised methodologies. And to give you an example of what these results look like, in a few example participants with normal hearing that I've tested in my lab, we again see this typical profile that I showed you that we call an inverted U shape, where we got this peak of importance around 1 to 2K, and then progressively less importance at the higher and lower frequencies. So this, again, is just saying that access to speech cues in that 1 to 2 kilohertz region are really important, and access to speech cues outside of that, a bit less so. Now, how does this change when you have a hearing loss? Well, some previous work has looked at individual differences in the speech intelligibility index. And the thing that's been introduced as a third proficiency factor is what the literature tends to call it. And what this is intended to model is individual variability in use of those speech cues and so now we can sort of customize this to look at what an individual's access to the speech signal is. I just used the word once. I'm going to keep using it throughout the rest of the talk. I prefer the term access over proficiency because I think proficiency implies some sort of active learned skill, whereas access I think is more neutral because it implies just how much of the speech signal do you have access to in any given frequency region. And we've done this in patients with hearing loss. And what we found is that there are really variable function band, or really variable individual differences in band access across patients with hearing loss. So on the left side of this figure, what I'm showing you on the bottom is an audiogram of a patient with a typical cookie bite shape, where you can see they have the greatest hearing loss around 1 to 2 kilohertz, and they have less hearing loss at the edges of the spectrum. So the thresholds are close to 0 at the lowest and highest frequencies. And then above that, what I'm plotting is the blue bars show this specific patient's access to speech cues in these different frequency regions. And what you can see here is that the function doesn't follow this inverted U shape. So behind those blue lines, I've got this gray shape that shows sort of individual variability in patients with normal audiometric thresholds. And relative to that typical gray shape, you can see that this person's access to speech cues in that 1 to 2 kilohertz region is really depressed when compared to that normal audiometric threshold function. And their access to speech cues, they're actually using those speech cues at the edge of the spectrum a little bit more than you would typically expect. And so that suggests that this function is sensitive to their specific configuration of hearing loss. We tried this with patients with a variety of configurations of hearing loss. So here in the middle, you can see a sloping high frequency hearing loss. And again, the green lines are biased so that they're using those low frequency cues that are more audible to this person more, and they're using high frequency cues less. And then we see the opposite in the person on the right-hand panel where they've got this rising configuration of hearing loss, where they're using those low frequencies the least, and they're using high frequencies relatively more to understand speech. So this by itself might not be that surprising because we know that the audiogram predicts a lot of individual variability in speech recognition accuracy. So showing that it also predicts band access is not something that's too surprising, but I think it's a really important validation of this methodology to say that our ability to measure band access on a patient by patient basis is something that follows something that we know it should. And the reason I think that's important is because then when we get into patients with cochlear implants, things are less obvious in terms of what the results actually mean. So here on the left panel, what I'm showing you is listeners with normal audiometric thresholds listening to vocoded speech. Now, if you're not familiar with this, a vocoder just strips out a lot of the fine spectral and temporal information in the speech signal and leaves you with sort of a coarse envelope of the spectral and temporal modulations that are there in the speech signal. This, we believe, simulates some of the signal processing that happens in a cochlear implant, although it's not analogous to cochlear implant listening. We just did it to degrade the signal in a way that we think is at least part of the signal chain when you're listening with a cochlear implant. And what you can see here in these listeners with normal hearing is, again, this typical inverted U shape that we expect, where there's a peak at around 1 to 2 kilohertz and it declines outside of that range. And individual differences shown as colored bars are fairly consistent across individuals in the study. So everyone who did this part of the study has a similar importance function. Now, if we look at actual patients with cochlear implants, here you can see people who use devices made either by Cochlear Corporation or Advanced Bionics. Two significant trends jump out to me. The first is that these plots kind of look like confetti, that these individuals who participated in the study dramatically vary from one another in terms of what speech cues they're using to understand what they're hearing. So this suggests that there's a great degree of heterogeneity in how patients have access to speech cues through whatever their processor and auditory pathway are doing with that signal. And this is potentially something that might be clinically actionable, because if we can identify patients that have poor access in a region we know is important, then maybe we can do something to modify the device to provide better access to those speech signals. The second thing that jumps out to me here is that the group average seems to deviate from what it looks like in the listeners with normal hearing. And you can see particularly around 200 to 400 hertz seems like the function has a really high importance on average across these patients. And so that suggests that we might have some problems with our assumptions about what do people actually need to be able to understand speech. Like the signal processing that happens in a cochlear implant is really unique to that sort of device, combined with the fact that you're using electrical stimulation to the auditory nerve directly. And so the frequencies that you use to understand that speech signal might differ from what we typically see in listeners with normal hearing. And so maybe there's an opportunity to revise how we think about providing access to the speech signal to make sure that we're really hitting these critical portions of the signal for these patients. And what I found in a little additional work that I'm not ready to talk about too publicly yet, is that individual variability in band access accounts for about 20% of word recognition outcomes in patients with cochlear implants. Now, that might not sound too much at first glance, but the current state of the literature in cochlear implants is that there are so many different factors that can affect these outcomes that this ability to use this one measure to predict about 20% of performance is actually pretty good relative to what we currently have in the field. And the fact that this is potentially clinically actionable means that this might be something worth evaluating. You know, a lot of factors, things like how long you've been living with a period of profound hearing loss prior to implantation, is not something an audiologist can ever do something about. Maybe from a public health perspective, encouraging people to get implants earlier when they start to hit the portions of severe hearing loss is something we could do. But once a patient comes in the clinic, there's not really anything you can do about it at that point. On the other hand, if somebody has poor access to speech signal, maybe there's something we can tweak in their device or how the device is processing the speech signal to improve access in that region, and thereby try and address some of these issues with access to the speech cue and improve speech outcomes as a result. Okay. So I think the natural question that follows from observing this variability is, why does it happen? What are the factors that affect access to speech cues across different frequencies in patients with cochlear implants? There are a lot of mechanisms that could potentially affect this. I'm going to focus just on two here. The first one is individual variability in the health of the auditory nerve. So we know that individuals who have a prolonged period of severe to profound hearing loss will often have variable survival of cells in the spiral ganglion. And as a result, this can lead to what are, commonly referred to as dead regions in the cochlea, where there just isn't a lot of auditory nerve left to be able to encode whatever speech signal you're trying to get through. It doesn't really matter how much stimulation you do. If there's no nerve, there won't be a response. There won't be a signal that's carried up to the brain. And we think this could be one reason why some patients might have poor access in specific frequency regions. The second thing that I think is likely to play a major role in this is electrical spread of excitation. So with a cochlear implant, you've got a series of electrodes that are in the cochlea. And what they're doing is they're emitting current pulses into the space inside the cochlea. And our goal is to get that electrical signal to the auditory nerve to stimulate, ideally, just a very narrow portion of it, so that we can convey good frequency specificity with our stimulation. But the problem is that electrical signals propagating through biological tissue are often very chaotic. And it really depends on where the cochlear implant is within that geometry of the cochlea, and also what's going on with the tissue surrounding it. Can all affect the pathway that that electrical current takes throughout that space. And as a result, you get dramatic individual differences in how far that current can spread throughout the cochlea. And that can vary from patient to patient and also from place to place within the cochlea within a patient. So these are the two factors that I've been looking at in my lab. And we have a really cool tool in patients with cochlear implants where we can look at the electrically evoked compound action potential, which is the synchronized response of the auditory nerve to electrical stimulation. So we can use the cochlear implant both as to stimulate the auditory nerve, but also we can use it as a recording electrode to record what's the neural response that results from that stimulation. And another thing that I think is really cool is that all major cochlear implant manufacturers currently have in their clinical software some ability to make these assessments. So you can measure the response of the auditory nerve using clinical software. The degree of control you have over this varies from company to company. Some let you do more than others with what you're planning to measure, but they all have it to some extent. So it's clinically feasible to make measurements using this technique to get an assessment of what's going on with a patient's auditory nerve. The way we often make these measurements, although there are several methods available, is called the forward masking technique. I'm not going to go too much in the details of how this is done, but just highlight a few important points. So when we stimulate with an electrical signal out of an electrode in the cochlear implant, we get two pieces of a signal that we can then record in response. We get the response of the auditory nerve, which is the thing we want to measure. And then there's also always an electrical artifact caused by the stimulation itself that we don't want to measure. And a while ago, some very smart people figured out a procedure for combining pairs of electrical pulses. So one is a masker and then one is a probe to extract just that portion of the signal that is the neural response. And what we can do is we can vary where those maskers and probes are within the cochlea to look at how much they overlap in terms of what they stimulate in the auditory nerve. And we can use this technique just to get the response of the auditory nerve to that stimulation. And we tend to get curves that look like this bottom figure here, with this downward deflection followed by an upward deflection. And we use the magnitude of that signal as our measure of how much the auditory nerve is responding to that stimulus. So these are two example waveforms I collected in my lab. At low stimulus levels, no waveform will be evident. Although I should say that doesn't mean that the signal is not audible to the patient. It's often the case that when you stimulate at relatively low levels, a patient will report hearing something, but the synchronized activity of the auditory nerve won't be large enough that you'll see the measurement here. So this isn't to say that there's no auditory signal getting through. It's just we can't measure the nerve's response at lower levels. And as we turn the level up, we get this typical shape where there's a downward deflection and an upward deflection in the signal we're measuring. And often what we look at is the amplitude of the difference between those 2 points in the signal, although some people also look at the timing or variability in that timing across measurements. So now looking at these amplitudes, what we can do is a relatively new technique that's called a panoramic ECAP. And the idea here is to try and measure the response of the auditory nerve to all combinations of masker and probe pulses to see how much they interact with one another. So in this figure, what you can see is this is the amplitude of the auditory nerve response to stimulation with a bunch of different probe and masker combinations. Along the diagonal, they're both at the same location, so you should get the largest measured auditory response because they overlap the most in terms of what region of the cochlea they're stimulating. As you move off of that diagonal, you're moving the masker and the probe farther and farther apart. So the amount that they overlap in terms of what they're stimulating gets smaller. And we can infer from the patterns in the data two underlying properties of the nerve and the electrode nerve interface. We can infer the magnitude of the response that I'm going to shorthand call health, and we can infer that spread of excitation within the cochlea. The math tends to look something like this. So we'll have some estimated parameters that we're trying to figure out of how responsive the nerve is at any given point. That's shown in the top of this figure. So shorthand, that's called health, although I want to caveat that by saying that's not representative of the entire health of the nerve or every auditory neuron, because we know there are a bunch of different things that can affect neural function. But this is just the magnitude of the synchronized response to that stimulation. And then that the responsiveness of the nerve interacts with the physical spread of electrical excitation within the cochlea, shown here in the blue, and those are convolved together to produce more complex patterns like this red line, where maybe you've got some good spread of excitation, but a region of poor responsiveness in the cochlea, and you can get more complex patterns that look like that red line. And our goal is to, giving those measurements that we actually have, fit the model to the data to best estimate what the health and spread is at every point along the electrode array. So as an example, this is a figure I showed previously. We can see that the brightest spot in this figure, the largest response we're getting from this patient, is around electrode 14. So we say the health of the response, or the nerve, is highest there. And outside of that, as you move more towards the base of the apex, you see that the function gets dimmer. So the response is smaller there. And so we say the health is lower on both ends of the electrode array for this patient. The other feature to note in these data is that near the apex here, you can see that this gray blob of data extends down pretty far in the figure. And so that suggests that stimulation from this electrode is spreading pretty far throughout the cochlea. And that gives us an estimate that there's a pretty large spread of excitation at the apex for this patient. Conversely, if we look at the base, you can see that this is a smaller gray area, and so that spread of excitation is smaller in that region. And so we can use this to make estimates of what's actually going on an ear-by-ear basis in terms of how their nerve is responding to stimulation from the cochlea. And what we can do, and what's still very preliminary in my lab, is compare individual differences in these ECAP measures to individual differences in access to speech cues at the corresponding channels in their device. And what we see is hazy. I really need more data to be able to make any sort of definitive conclusions here. But there seems to be a trend for some patients for the spread of excitation within the cochlea to be predictive of their access to speech cues. We really need more data to confirm that, but it suggests that maybe there's something here that'll relate to band access and ultimately speech recognition outcomes that we might be able to use in the clinic. So to summarize the first portion of my talk, we know that individuals with hearing loss vary in their access to speech cues in different frequency bands. So we have results in listeners with hearing aids and in cochlear implants. I'm also doing some work looking at how this works, not just in adults, but also in children. So we might be able to use this in a lot of patients with hearing loss to ultimately try and identify mechanisms that are impairing their access to speech cues, and then use that to revise the design of their devices to give them better access to those cues. And this is more speculative, but I think there's promise in patients with cochlear implants to use these compound action potential recordings to yield additional diagnostic information that could help an audiologist make decisions about how they're going to adjust the device to improve outcomes. All right. The second piece of my talk I want to focus on now is the cognitive elements that affect speech recognition. This is a model that is pretty commonly referenced in the cognitive hearing science literature. It's called the ease of language understanding, or ELU model. And I'm not going to go too much into the details here, but just to say that it places speech recognition as this process where you've got incoming sensory information that you're trying to make sense of using your cognitive abilities and linguistic knowledge. So as speech signals come in, we often think of these as auditory signals, but the model acknowledges that vision sometimes plays an important role here too. What you're constantly trying to do is you're trying to figure out, as the speech signal is unfolding, as you're listening to every word somebody is saying, your brain will briefly activate a cohort of items that could be plausible interpretations for what that word is going to be. And then as the word unfolds over time, you winnow down that decision space from among those competing interpretations of what it is you're hearing. Assuming everything's going well, you ultimately select a single interpretation of what that word actually was. If the signal is degraded, you might be left in a case where you're uncertain about what exactly that was. But that's where the second larger loop in this model comes into play, where as you're listening to speech, what you're also doing is remembering what came previously and using that to fill in missing pieces of information, chunk related pieces of information together into larger concepts, often to get the semantic gist of what somebody's talking about. Even if you don't remember every word, you might remember the core ideas of what was communicated, and also to make predictions about what it is somebody's going to say as a means of facilitating real-time speech recognition. So we think there are potentially two mechanisms that are really important here. At this small lexical access level, variability in your ability to quickly select an interpretation of what you're hearing is likely to be important. And then at a broader, more conversational level, your ability to remember what it is you're hearing and use that to make inferences about missing pieces of information is likely to be really important in that context. So to start, one of the experiments we did early on this is just look at what predicts word recognition in our band access tasks. This project was done with a former trainee of mine, David Yeager, who's currently at University of Louisville. So in this project, we test for predictors of recognition accuracy in our filtered word recognition tasks. So we had people do these band access tasks, and we also had them do a few different cognitive tests just to see if anything related to their performance in this test. And also, I forgot to mention earlier, this what I'm going to show from here on is all young adults with normal hearing, so we can really precisely control the auditory input to really focus on what's the cognitive side of the problem. I won't go too much in the detail of all of these tasks, but I will highlight one task that measures attentional control, ended up being a really strong predictor of word recognition for this study. The task we used is the flanker, which is pretty widely used to assess attention. This is a very simple task where you're shown a set of arrows on a screen. In this case, it's 5 arrows, and your goal is just to pay attention to that middle arrow and tell me which way is it pointing. Is it pointing to the left or the right? And you've got 2 buttons usually on a keyboard, and then you press left or right, depending on what that middle arrow, which way that middle arrow is facing. So it's a really simple task. And overall reaction time on this task doesn't seem to relate to speech recognition accuracy. What does matter is what happens when we introduce a little bit of conflict in what people are sensing. And here you can see it's still the same task. That center arrow is pointed to the left. So you should push left on your on the keyboard in response to that. But now those flanking arrows are pointing in an opposite direction. And that's just a little bit of sensory conflict that slows people down. And what we found is how much you slow down as a result of that sensory conflict seems to be a pretty good predictor of recognition accuracy. So the people who were slower when we introduced the sensory conflict also tended to be worse at the speech recognition task. And we think what's happening here is that in both cases, you've got some incoming sensory information that's activating sort of an automatic response to that signal. In the flanker task, it's assessment of which direction the arrow is going. In the speech recognition task, it's activating a cohort of competing interpretations of what that word could be. And we think your ability to resolve that conflict efficiently affects performance in both tasks. And I think this is probably pointing to some sort of general attentional mechanism, because here the speech recognition task is an auditory task that is explicitly verbal, and the flanker task is a visual task that is nonverbal in nature. So we think it's fairly general, although I need more research to follow that up. This is one study in adults where admittedly the number of participants is relatively small, but we also found similar trends like this in children in a study in one of my collaborators' labs. So I do think this is a real finding, but a little more work is needed to figure out what exactly is the underlying mechanism here. But so we think attentional control is important when you're trying to understand individual words. The story changes a bit when we're looking at sentence recognition instead. This is work done by a former trainee of mine, Michael Berry, who's currently a medical resident at University of Kansas. And what we tried to do here is to look at vocoded sentences. Now, these are longer, complex sentences, and see what predicts your ability to understand those. We did a bunch of tasks that I'm not going to go through all of these again. The 2 I'm going to highlight are the 2 verbal memory tasks we used in this study, because those are the ones that ended up being important. So we tested our participants with vocoders that were 3 different difficulties. So with a vocoder, you can control the number of spectral channels in the signal manipulation, with the idea being that as you reduce the number of channels, you're reducing the amount of information the person has access to, and so performance gets worse. So you can see going from 16 channels in the vocoder to 4 channels in the vocoder, the average group performance declines pretty sharply. But what you also see is individuals in this population, so every line represents one person in the study, tend to be consistent in their performance relative to the group. So people who are good at the task in the easy condition tend to be good at the task in the hard condition. And conversely, people who are poor at the task in the hard easy condition are also fairly poor at the task in the hard condition. So that suggests that there's a single underlying factor that affects performance across task difficulties. And we also found that measures of verbal memory were predictive of that. So the 2 specific memory measures we used in the study were a digit span, where we played people a list of digits and asked them just to repeat those back in order. Performance varied across individuals. You can see here that at our longest list we tested, which was lists of 9 digits long, people varied from getting 3 or 4 digits correct to getting almost the entire list correct. So there's substantial individual variability in performance in this memory task. And that performance was also related to their ability to perform a serial word recall task. Now, these are words that have no semantic relationship to one another. It's just an arbitrary set of words. We play you, and your goal is still to repeat them back. You can see here performance is variable across individuals as well. And I'm not showing it, but there's a really strong correlation across these tasks. So individuals who are good at the digit span task also tended to be really good at the word span task. And so we combined performance across the memory tasks into a single z-score on the x-axis. And we also combined performance across listening conditions in the sentence recognition task on the y-axis. And we saw that, again, there are really strong correlations between cognitive performance and speech recognition, even in young adults with normal hearing. So what we think this means is that when you're trying to process longer and more complex stimuli, what's dominating your ability to do that task is your ability to remember sequences of speech information and potentially infer missing pieces of information from that memory. So a problem I want to address that is something that I've become increasingly frustrated, even in my own work with, is that correlations between performance and 2 tasks can be really ambiguous. Often the way we talk about things is that we measure task A and we design task A to measure one specific factor about a person. And we measure task B, some outcome that we're interested in. And if they're correlated, we say whatever that underlying factor is, that must be the thing that's driving the correlation that we observed. But unfortunately, we know that's not true, especially when we're talking about cognition. The dimensions along which people vary are not entirely independent of one another. It's often the case that things like your ability to control your attention or your ability to remember sequences of information are correlated across individuals. And it's often the case that tasks are not pure measures of one thing. So you can think in a speech recognition task, I made an argument that attention matters in some cases and memory matters in some cases. And the reality is it's probably always both. If you're failing to attend the speech, it doesn't matter how good your memory is. You're probably not going to do well at the task. And conversely, if you've got really solid attention, like you're very on task, but you do have some memory impairment or something like that, you're also probably not going to do well at the task. So I want to try and move the field beyond just looking at specific assumptions about what these tasks actually measure and go to more of a modeling approach to say, if all these things, if a bunch of different measures are believed to measure the same underlying thing, they should relate to one another in a mathematical framework that we can test. So we put together in my lab a more sophisticated modeling approach to look at performance in some of these tasks, with our goal being to try and estimate not just are things correlated with each other, but what's the best explanation for why those correlations happened. And so the way we frame these things is by looking at a set of underlying factors. Those are the things that we actually want to measure about a person, and acknowledging that those factors will affect performance in different tasks to variable extent. And we do this on a trial level because that gives us a lot of granularity to say, how well does this explain the data? And so we model most of our tasks in a mathematical framework that basically just says, for most of the things we do, whether it's sentence recognition or memory, there's a given probability of getting X items, like say X keywords correct, out of a total of N possible items that are in the sentence. So like X keywords correct out of a total of N keywords might be a way we would look at this. And then we model the probability of being your accuracy in that task as arising from the difficulty of the task, acknowledging that not all tasks are equal, and especially if we're explicitly manipulating like the number of vocoders and the number of channels in the vocoder, we can control that task difficulty. Individuals, we believe, vary on their ability to do these tasks, and the extent to which that variability in their ability to do these things will affect different tasks to a different extent. So we use the statistical approach to try and estimate what each of these factors are to make some inferences about what's the underlying structure that explains the performance in all of the tasks that we're having people do. So the way we tested this is in a study I did with two trainees that I've worked with, Chantel Cannon, who's currently at Ohio State, and Gianna Doria, who's currently a medical student at Creighton. And what we did is we tested people on a variety of memory tasks and also a very mentally demanding speech recognition task. So in the speech task, we had complex sentences in a two-talker masker that were then vocoded. So trying to really make this task very mentally demanding for people to see sort of what results we get. And what we found is that all three of the memory tasks we used were predictors of sentence recognition accuracy, and they were all correlated to one another, which again suggests that there's some underlying commonality to performance in these tasks. And I'll gloss over the modeling a little bit just to give you the core results. What we found is on the memory task, the best explanation for performance in these tasks was a model that had two distinct factors in how people were performing. One factor that I'm highlighting the boxes in purple seemed to be general across tasks. It affected performance in all three of these memory tasks. And then a second factor seemed to be more specific just to the digit span task, and specifically accuracy in that task at longer list lengths. So we think that maybe remembering digits is sort of a unique skill that's distinct from general verbal memory. And intuitively, I think this makes sense. The idea that language is not the same as your ability to manipulate numbers, that those are distinct dimensions for people. What we found is looking at individual variability in these two factors is that they were pretty independent of one another, and individuals were all over the place in terms of performance on these two dimensions. So every one of these circles represents one person, and then the clouds around those circles are the uncertainty in those estimates for each person. So these people meaningfully vary from what in their performance on these tasks along these two dimensions, this one general dimension and this one digit specific dimension. Now, when we look at how that compares to sentence recognition, what we found is that that general memory factor seemed to be very, very strongly associated with their ability to understand these sentences in this complex listening condition. They're so strongly associated that actually there was a one-to-one mapping in the model. I don't think that's necessarily true, that these are exactly the same factor, but they're so highly correlated that at least in the data that I have available, we couldn't dissociate the two at all. The model is saying that these are the same factor effectively. So I think that makes an argument that general verbal memory ability is really strongly associated with speech recognition in more complex listening conditions, which are likely to be the conditions that your patients are have the largest complaints about, because those are closest to what we consider to be real world or naturalistic listening. Like, your patients don't come into the clinic complaining that they can't understand one word at a time spoken in quiet. They often complain that they can't understand their friends in a restaurant. And so acknowledging the fact that there's cognitive factors that we might not be able to do anything about in an audiology or an ENT clinic is something that I think is important for managing expectations in patient care. So my current perspective that I'll end on is just reiterating that the properties of the stimuli seem to moderate your ability to access speech cues in that signal and the cognitive demands of the speech recognition task itself. So what I showed you is that acoustic or auditory degradation of the signal can affect your ability to access speech cues. And the semantic content and complexity of that speech can determine what aspects of cognition are playing a major role in your ability to understand that speech. So my takeaway from this research as it stands is that we really need to be able to design assessments and then modify clinical devices to consider both the fact that your patients are going to vary in their access to these auditory cues and also their cognitive ability to make use of the cues that they're being given. And I think being able to separate these things will really help us focus on the things that we can modify in the clinic and precisely identify what those things are on an individual basis. And also acknowledge that there are some things that are probably beyond our scope of practice that we can't address with the device, which, even though it might be disappointing to a patient that we can't give them a better outcome, being able to counsel them and manage expectations is really important for satisfaction with the device and continued use of assistive devices. So being able to give people good expectations as to what they're likely going to be able to achieve with their device is still really clinically helpful. So with that, I would like to acknowledge my many collaborators and particularly acknowledge the trainees and assistants who collected most of the data that I've shown you today. And with that, I would love to take questions from the audience. Thank you very much.